Hi, I'm Anna Poslednik, and I am a graduate student at the Virginia Institute of Marine Science, or VIMS. My research at VIMS focuses on the ecology and the evolutionary biology of the oyster parasite, commonly known as dermo. Here I am presenting a lesson plan that's intended for high school biology classrooms that focuses on understanding a laboratory technique that I commonly use in the lab to diagnose diseases in oysters, known as PCR or polymerase chain reaction. This lesson plan, entitled Serial Killer, Diagnosing Oyster Diseases Using PCR, starts off with a murder mystery where students learn about oysters and are given a scenario where an aquaculture farmer has experienced a mass mortality of her oysters. Students are then asked to brainstorm in small groups possible reasons or murder suspects for this mortality event. In the first activity of this lesson, the students work in groups to analyze different graphs and data taken at the crime scene on this worksheet. After analyzing all of the graphs and tables and answering a few short questions, the students will then hopefully decide that some sort of disease has caused this mortality event. Following the first worksheet activity, teachers are encouraged to present a few slides which are provided about the different parasites and that may be causing the murders, as well as what a PCR is. Then students will work in their small groups to model a PCR using cereal and pipe cleaners as their DNA. So this activity requires teachers to prep one double-stranded DNA sample per group, which is made up of 20 Fruit Loops on 2 pipe cleaners each. And teachers can use off-brand Fruit Loops as well. And these cereal are arranged in a specific color order, and they also have to prep 3 sets of primers. Each set of primers includes 2 pipe cleaners with 5 cereal Fruit Loops in a specific color combination. So this is 1 set of primers. Students will then use these materials to understand each step of a PCR, from denaturation, the splitting of the double-stranded DNA, to annealing, the binding of the correct primer set of the 3 provided, and elongation, the addition of nucleotides or cereal to the end of the correct primer set to complete their DNA strands. Students will also need to figure out which primer of the 3 provided matches their sample DNA by following this color matching scheme, where blue nucleotides or cereal will bind to green nucleotides, cereal, and orange cereal will bind to yellow cereal. After identifying the correct primer set for their samples, teachers can decide whether to have students perform 2 more rounds of PCR by providing 2 more sets of the correct primers to each group. Or to save on time and resources, teachers can also opt to hand out a products of 3 rounds of PCR handout with a photo of all of the DNA strands after 3 rounds of PCR are complete. Using their DNA strands or the handout, students will then complete a worksheet with a few short questions and then run a gel of their PCR products by filling in this diagram. With their gel diagram, they will then be able to identify the oyster serial killer in this lesson. So thank you for watching, and I really hope that you consider using this lesson in your classroom.